The descriptive word sexual is attached to seeds propagation because of the union of the male and female sexual gametes, which the process is called fertilization, is a requisite in the production of the seed or in the development of a new plant from a spore. Compared to vegetative or asexual methods, sexual propagation with the use of seeds has the advantage of massive production of seedlings and rootstocks. However, the seedlings may not be true to type because both the maternal and paternal genes contribute through the process of fertilization to the genotype of the embryo in the resulting seed. Meanwhile, the transmission of traits is only of maternal origin, so that the ensuing plant that it develops from the seed exhibits the characteristics of the seed-bearing parent only. They could adapt to possible changes in the environment and tend to survive and produce next generation.
And now I want to talk about the uses of seedling in propagation. There are numerous applications for seedling in propagation, including mass propagation, rootstock for sexual propagation, and plant breeding. So, what are those? Mass propagation is an effective and cost-effective form of dissemination. There are numerous benefits to using seeds in the mass propagation strategy, as long as genetic diversity is kept below acceptable limits. It can be created in large quantities over lengthy periods of time, transported all across the world, and mass spread in a highly effective manner. For rootstock, seedlings are often utilized to supply rootstock for vegetatively propagated plant varieties, particularly for grafting and budding. Seed materials that have been carefully selected can be produced consistent, vigorous, virus-free seedling plants at a lesser cost than similar vegetative method. So lastly, for the plant breeding, the growing seedlings in this method is the most essential method of generating the new cultivars because of the heterogeneity caused by chromosomal segregation during sexual production.
I will explain about self-pollinated species and cultivars. As we know, self-pollination is a process of transferring pollen grain from the anther to the stigma in the same flower or different flower but at the same plants. However, most species naturally cross-pollination. So how could it happen? It happens because these species have specific mechanisms that prevent self-pollination. As we can see in the slide, they have 5 mechanisms that prevent self-pollination. The first one is dioecious. Dioecious plants have either male or female flower in each plant. The second one is monoecious. Monoecious plants have pistil and stamen that develop in different flower but on the same plant. For example, is corn. The third mechanism is dichogamy. Dichogamy is a condition where the anther and the stigma are mature at the different time. There are 2 forms of dichogamy. The first one is protogeny and the second one is protandry. Protogeny occur where the stigma mature before anther, where the protandry is vice versa to the protogeny. For example, is avocado plant. Avocado plant have both protogeny and protandry variety. Next is gametophytic sterility. Those plants have specific genes that stop the normal formations of male or embryonic reproductive structure. For example, nuclear gene can cause sterility in the male reproductive structure. As a result, functional pollens could not be produced. Lastly, incompatibility. There are 2 main types of incompatibility, which are heteromorphic and homomorphic. Heteromorphic is associated with differences in floral structure. As we can see in the slide, they have distylic and tristylic. Both of them are different in style and anther. Whereas the homomorphic is not associated with differences in floral structure.
I will discuss about cross-pollination. So what is cross-pollination? So cross-pollination is when pollen grain from one plant are carried to another stigma of different plant. So stigma is the female part of the flower which that will germinate and fuse together with the male gametes. So there are three ways of cross-pollination, which is insect pollinated, wind pollinated, and bird pollinated. So the three ways of cross-pollination are totally different. So same goes to their structure. As you can see, the insect pollinated, the pollen grain are larger and come in small amount with a hook. This is for the pollen to be able to attach themselves to the insect. So the flower must be bright and scented in order to attract the insect toward the flower. For the wind pollinated, the production of pollen grain is enormous and light with smooth surface. This is to make sure the pollen grain be able to blow away far by the wind. So the stamen is long, protrude above petal, and the stigma is long, feathery. So for bird pollinated, the process is quite similar with the insect one, which the birds will brought the pollen far from their parents. So the common bird in cross-pollination are human birds, honey-eater, sunbird. Usually the flower is in yellow or red color, which attract the bird attention.